Then coming to the next topic, that is microfossils. Okay, microfossils are very tiny fossils which are sometimes not even visible to naked eyes. Okay, so microfossils are tiny fossilized remains or traces of microscopic organisms. Remember, these are microfossils means these are microscopic organisms, cannot be seen through naked eyes. They are often studied for their significance in various scientific disciplines. Okay, they typically measure less than 1 millimeter in size. So less than 1 millimeter in size, we call it a microfossil. Okay, just like microbes, microorganisms, there are microfossils. So which kind of microfossil we will find? Obviously, the soft-bodied microorganisms that died, we won't be able to, they won't get fossilized easily. So only microfossils that we get that were actually uh, that lived there, that lived at that time, and that had some kind of hard covering, so that the fossilization process can take place, and those can be fossilized. So all the microfossils that we'll study today, some hard parts were there, maybe the full body fossils, or some maybe some soft organism had some kind of strong teeth. Okay, so those teeth can be preserved, and those can be fossilized, and we can study those now. So I was talking about conodonts. Conodonts, I'll show you one by one. Okay, so I'll show you the introduction to microfossils. So A is actually the calcareous mineral world. So those microfossils which had a mineral world, that is a calcareous mineral world or calcium carbonate mineral walls. So so it will look like this. You can see this was a microfossil which lived inside a calcium carbonate wall. This is actually a foraminifera. Okay. The first one is the most important one is the Foraminifera, which is actually a single-celled marine protists. Remember, it's a protista with calcareous or agglutinated shells. Agglutinated means collected shells, different collection of different kinds of shells. Benthic to planktic, it can be benthic. That means it can be a bottom dweller. That means lived on the ocean bottom, or it can be planktic. That means it's a floater. Okay, it can float on the ocean water. So both can be found, benthic and planthic, and because this benthic and planthic ratios are very important to understand the different paleobathymetry. Paleobathymetry means the old depth of the ocean at which the different organisms were found. That is paleo depths of the ocean. It's called paleobathymetry. Is a very good indicator. Benthic by planthic ratios at some areas will give you the idea of the paleobathymetry of the area. Okay. Then significance, so used in paleoclimatology, paleoceanography due to their abundance and sensitivity to environmental changes. Okay, so we'll talk about foraminifera later on. Okay, we'll talk about ostracoda also. So all those microfossils. This is just a introduction to different kinds of microfossils, and we'll talk about all those things one by one later on. Okay. So foraminifera it was done. Foraminifera, I'll show you a picture of foraminifera, how they look like. So this is actually a foraminifera. So foraminifera, or you can shortly say it as forams. Okay, forams. These are different kinds of forams. Okay, mostly those forams which have spots on them are actually floaters, and those forams which are actually very large, globulated like this, are actually benthic forams. Benthic, and those which are like this, spotted and have voids in them and mostly thin, are actually the planktic forams. Why? Because benthic forams do not have to float, do not have to swim. That's why it can be heavy. Whereas planktic forams need to be very light, need to have some pores so that it can float on water. That's why they are like that. So these are heavy forams, benthic forams, round, uh, and these are planktic forams which are actually thin, disc shaped, and contains lot of small valves or small cavities like this, you can see. Okay, so these are different kinds of forals. There are many kinds of forams. There are maybe I don't know, maybe some 50 to 60 kinds of forams. Okay, so these are different kinds of forals. There are many kinds of forams. There are different kinds of forals. There are many kinds of forams. There are many kinds of forals. There are many kinds of forals. There are many kinds of forams. There are maybe some 500, 400 types of forams are there. I don't know the exact count, but I'm just saying there are a lot of different kinds of forams are there, and each type of forams will give you some facts or some information about some, or like each types of forams have some application in geology. Okay, so forams are done. Next are the coccoliths. So coccoliths, or you can say coccolithophores. Coccolithophores are unicellular flagellate. Flagella means it contains a flagella. Flagella is just like a ribbon shaped organ. Okay, flagella. Algae organisms living in photic zones. So these are actually algal organisms. Okay, algae. They are nanoplanktonic, and cell wall is covered with calcareous plates. So these are actually calcareous plates. Okay, so we'll talk about these plates, and their study helps scientists reconstruct ancient climates, track evolutionary patterns, and assess the health of modern oceans. Okay, again I'll show you. I go call it the four. Image one. This one, yeah. So this is a coccolithophore, and these are the discs. These are the discs of calcium carbonate. Okay, this is a separate disc, right? So you do not have to understand the morphology of coccolithophore because that is not the main thing. Sorry, but like the applications of coccolithophores are very important. Okay. Okay. Then there is the ostracods. Ostracods are typically enclosed in a two-part. Okay, in a two-part hinged or bivalve shell made of chitin. Chitin is another material. Okay, chitinous. So remember, ostracods are actually chitinous. They generally range from less than 1 millimeter to a few millimeters in length. Found in diverse aquatic environments, include marine. It can be found in marine or salt water. It can be found in fresh water like lakes, rivers, and even damp terrestrial habits like swamps. Okay, valuable in biostratigraphy due to their abundant fossil record. Okay, since ostracods of different types are found in different areas around the world, that is why again it is used for biostratigraphic correlation. So I'll show you ostracod next. This is an ostracod, you can see. So this is actually a carapace. This is actually the carapace of the ostracod, made up of chitinous material, and this is the soft body part of a ostracod. So this is an ostracod, and we mostly get to see the hard part covering getting fossilized, where the soft part of the soft body is actually gets withered or gets decayed. Okay. Again, the morphology of the ostracod is not important, but the environment and application of ostracods are more important. Okay. So just to ease you guys out, that only the morphological parts of a foraminifera will be important for exam point of view. Others, only the application will be necessary. Okay. So only foraminifera you have to study the morphological parts. Okay, different morphological parts, and also different ornamentations in foraminifera. But other microfossils, you have to know only the uh, type of material that it uses for the covering, and the application part mostly. Okay, Coccolithophores, ostracoda, conodonts, diatoms, all those things. Okay.